Couple papers, recent papers from my group. I'm at Colorado, and we're going to talk about this method called Magma-Seq, which is this little guy right here. So I like to start with acknowledgements in case I run out of time. This work was really done by a few students in my group, most importantly Monica Kirby, who's right here. She's kind of the lead author on a couple papers I'll present on. For the grad students in the audience, have interesting pictures for your PIs. Okay, so the premise of this talk is that AI can solve biophysical problems. This is Demis Hassabis. There's a picture I took of him at the Nobel lecture in chemistry in December in Sweden, and he won the Nobel Prize on this. So of course this premise is true, right, from AlphaFold. But the key here is you need enough experimental measurements. And so in this lecture, he gave a slide where he addressed what makes a suitable problem for AI. And really, there's kind of three criteria. The first criteria is there's a massive combinatorial search space. And I think most of the problems that people are interested in this room, that box is checked. The second is a clear objective function or metric to optimize against. And really defining that objective function was easy for protein folding because it had been well stated for 30 years, well posed. And I would argue that really defining that objective function is important, kind of the most important things that we can address academically in the next few years. And then three, lots of data. You need lots of data to optimize against. Okay, but here, what the point I'm trying to make for this talk is that you need these experimental measurements. So what I'm interested in, I've been interested in for a long time, is solving protein ligand molecular recognition. And so what that means is really thinking about the biophysical principles in terms of free energies of how proteins interact with different molecules like proteins or nucleic acids or small molecules. We've been at this since I was a postdoc. We took a hiatus for a while, and we recently kind of come back to the field because the technologies right now are really outstanding. But I would argue, and I think most in this audience, if not everyone in this audience would agree, that this is an unsolved problem, right? You generally can't design any kind of protein for any surface with 100% accuracy and predict that free energy. So I think this is going to be a grand challenge in the next decade or two, but I think a near-term tractable challenge, and I think the domino that may fall first is really the problem for antibodies. So you can pose this for as such, like for an arbitrary biomolecular surface, can you discover a set of specific antibody binders and quantitatively predict their affinities. And now I know some of you in the audience are thinking, well, you know, antibodies, those are boring. Let's go on to the next things, and which I would say that they're not. Actually, antibodies are quite interesting. So the question I really want to address in this talk is thinking about the scale of the data that's needed. So Charlotte Dean at Oxford has spent a lot of time, her career, kind of working on antibodies, and she kind of published a preprint a couple years ago that should see press in the near future, and really just asking how much data you actually need. And kind of in this community, you guys think typically pretty deep, right? You take one or two or a few proteins and really scan and make tens of thousands of mutational data points for individual proteins. And instead, her argument is that you really need a wide approach. And what that means is you need a few hundred mutational data points, but you need to spread them across really diverse sequences. And so the order is really gobsmacking. It's like 10,000 or 20,000 antibody antigen complexes, but you only need a couple hundred data points for each of those complexes. So we really have been thinking for a long time about how to build an experimental platform to really meet that challenge. And so you really need kind of four things. You need quantitative measurements. You need to be able to have scalable, so you need to be able to scale for multiple antibodies in a single experiment. You need to have internal controls in this, and then you also need to have a fab format, and I won't talk about this because I don't think there are many antibody aficionados in the audience. So what we did is we really have iterated over previous technologies that our group and others have built over the last 2 decades or so. And the kind of moving parts in this is pretty simple. We take kind of a antibody fragments, so there's a heavy chain and a light chain, there's a variable region for those. And then we integrate them in a plasmid where it contains a molecular barcode. We transform that into yeast and display that antibody fragment on the surface of yeast, and then we go and use fluorescence-activated cell sorting to go and discriminate things that bind antigen that have fluorescently conjugated versus things that don't. We use Illumina sequencing of the sorted barcodes, and then we use a maximum likelihood estimation algorithm to go and determine KDs quantitatively. Okay, there are three big technical challenges to solve. We solve them. I'm not going to go into the specific details of solving them, except for one in particular, which is determining those KDs from sequencing data. So the work we do is really a subset of sort-seq. So for those in the audience who really don't care, you can look at your phone for the next slide or two, and then I can tell you when to pay attention again. For the sort-seq aficionados, pay attention. Okay. So for that, that determination of dissociation constants from sequencing data, what we do is pretty simple. We take fluorescence-activated cell sorting at different ligand concentrations, and what we do is we keep the population constant in the gate that we sort at. And so what you can see as this population increases with increasing ligand, the fluorescence intensity of those, we increase the gating threshold to do that, okay, to keep that population constant. What we get out is we get out sequences for each bin that we sort at, and then we can convert that to a probability. And so what we get is a probability vector for each set of labeling concentrations for each variant in the population. And then the key is that you can back out that mean fluorescence intensity, but we don't do that. I'm just showing you if you do this over many different ligand concentrations, you can have a titration curve where mean fluorescence versus ligand concentration, and the half maximal is kind of your KD. The key that really worked, and this didn't work for a long time, but what we figured out is that we use the maximum likelihood estimation on the probability vector themselves, not the inferred fluorescent values. And so that's kind of the key to work. Okay, so that was the technical bit. For those not paying attention, you can come back now. Okay, so this works, and this is great, and it's fantastic. And what we get out is we get kind of a log likelihood profile, and then the maximum of that is going to be the most likely value for that KD. We also get confidence intervals. And then what we can do is we can group these by barcodes for individual variants, and they kind of all map within that distribution. And then those error bars are the 95% confidence intervals for each barcode that's called. And you can see that the barcodes map kind of that distribution to go and get our KD value. We can do this. We do true biological replicates for this demonstration in this paper, where we sorted multiple antibodies together. We get reasonable agreement between KD and reasonable. What I mean by that is that the unsigned error on this is going to be less than 0.3 kcalamole. So this is actually pretty decent quantitative biophysical measurements. So we were really excited about this. About 2 years ago, I got together a large consortium of deep learners, people who do cryo-EM for a living, people who discover antibodies, and we asked the government for $100 million, and they said no. And so they took us seriously, which is nice, but they still said no. And we really wanted to go and ask how we could use this technology and really go and address kind of questions in antibody development as well as antibody engineering. I'll show you one example of that. So one question that we could address with this technology is really ask the question why antibody development is largely repeatable across human populations. So to address that question, or address the premise, I need to give you a little immunology. So if you can go back to kind of your immunology in undergraduate or graduate training, you come to this paper from Herm Eisen, where in this experiment, what he did is he took a hapten, so a small molecule that these rabbits have never seen before. He injected that hapten, and then what he did is, at time course series for individual rabbits, took out bulk sera and measured the antibody response. And what he found in this experiment is that the KD of that response, or the dissociation constant of that response, decreased after that injection. And what we call this now is a process of affinity maturation for antibodies. And so antibodies, when they go and are exposed to naive antigen, the antibodies in the form of these B-cell receptors will go and make somatic hypermutation to go and identify mutations on those antibody sequences which bind tighter to that antigen. What we have on this is that if you think about the math on this, and you think about the number of sequences you can generate from a human, and you think about where you're starting from, which is kind of 100 micromolar or higher, the chances of seeing the same antibody sequence in different humans when you go and are exposed to the same antigen is vanishingly small. So you can kind of just run the combinatorics of that. What turns out is that the same antibody sequences tend to be elicited in the same people, and those tend to be the immunodominant response. So what that means is the overwhelming majority of the antibodies are shared across individuals for naive antigen simulation. This was really well understood, appreciated in kind of 2020 and beyond, when people were just kind of mass individual B cell sequencing of patients infected with SARS-CoV-2 or vaccinated. Okay, and so we want to ask why this antibody development is largely repeatable across human populations. And so what we did is we used our technology to go and recreate the likely development trajectories of these antibodies. And the experiment is really simple. We take the inferred precursor sequence, so without any mutations, and the mature antibody, and those mutations are marked by X. And then we just make all possible combinations of those mutations. So the library size is 2 to the n, where n is the number of mutations. And then we go and record the quantitative dissociation constant for each variant in that library. And then from there, we can recreate the development trajectory, the likely development trajectory for those antibodies. We thought that maybe there was differences in antibodies and how they developed. So if an antibody is permissive, then any mutation can occur in any order, and all mutations are equally likely at each step in the development trajectory, versus kind of a more restricted development, where antibodies accrue a given mutation at the first time step, and that mutation leads the way for a second mutation that's important that it accrues. And it really has to go and skip the particular set of stones, which makes development much less likely. So I'm going to show you a lot of data. I want you to just look in the left-hand corner for this next slide first. So this is a subset of the data that we generated, where we're looking at each column here is the development step along the antibody. Is this working? Yeah, it's working. Okay. So for example, this antibody 4A8, this development step is the first mutation, and then these rows represent the mutations in the mature sequence. And what you can see is that the colors and the numbers are about the same at the first step, they're about the same at the second step, they're about the same at the third step, and so on and so forth. And you can look through this, and there are a couple antibodies where a mutation is more likely to occur initially, but really the order and the color and the shading is pretty consistent throughout. So what this really argues is that the antibody development, forget what I said about antibodies not being boring at the beginning of this talk, antibody development is largely boring and predictable. And mutations can happen in any order, and there are really only 1 or 2 key mutations there. So this doesn't really address why these public antibodies are repeatedly elicited, but when we look at the data and we map the KD, the initial KD versus development step, we can really see a difference. This is just a subset of the data. This is just for the SARS-CoV-2 data, and what we're looking at is public antibodies versus non-public antibodies. And what we see is that the main difference in this development trajectories is that these public antibodies are wealthy, and they're starting on third base. That's a baseball analogy. Maybe you guys won't get it. They're rich, and they get richer. They only need 1 mutation, and then they're kind of the same affinity as a mature antibody. And so what this argues is that the immune system, the adaptive immune system, really has pre-encoded specificities for certain antigenic surfaces. Okay. So I'm going to end there. This is really just a biophysical view, and really rethinking kind of this Eisen model, where you kind of start at this really, really weak affinity and mature throughout. But it turns out for many of these surfaces, if you're hard coding kind of higher affinity up front, you can get an immunodominant response. Okay, so in the time I have left, back to the 10,000-foot view for solving molecular recognition if we don't have $100 million. The first place is really finding ways that people have gone and evaluated antibodies at scale. And there have been plenty of campaigns over the last 2 decades or so to do this at a proteome-wide scale, some public, some private. The challenge is that finding that data and getting that data is hard. And so right now, I'm on sabbatical at the Sanger Institute. And there was the PI, John McCafferty, who's really, really well respected and well known in the antibody community, and he developed a similar data set 20 years ago. I actually tried to find it. I couldn't find it externally. There's a 404 message for the publication. I asked the IT at Sanger to go and look at the tape, and they were like, "Yeah, no, it's impossible." So this data exists, but finding it is hard. But it's cheaper than asking the government for $20 million to recreate that data set. The second is structures. So industry is really focused on building, buying cryo machines, and so we can go and solve structures at scale and being able to do this. I think there are better ways and faster ways to go and determine structures at scale. And then the third thing is mutational scanning. I think the big challenge with this technique that I'm presenting is that you have to do QC and you have to express and really test each recombinant antigen that you use in the machine. So if you can get away from that, then you really can scale this much faster. So that's kind of what I'm doing on sabbatical right now at the Sanger, and hopefully I'll be able to present this in a couple years. So really, the key take-home for today, we think antibody molecular recognition can be solved. We need the data to do it. We developed a program in order to solve this challenge. We learned how natural antibodies develop using this technique, and I'm bullish on the future of really marrying experimental data sets with deep learning, and I'm happy to be at this meeting to have a lot of like-minded people here. Thank you so much.